today. We might go over into lab time some. So just FYI, but chapters 15 and 16 are absolutely like key. They are fundamental, not just for the course, but just for understanding like healthcare and stuff like that. So understanding your immune system, what is inflammation and what's triggering like the repair of our cells and things like that. A lot of that stuff is tied in with inflammation, and the innate immune system as well as the acquired immune system. So that's what we'll be getting into today. So I wanted to take the time to actually lecture over it, since I know a lot of us don't actually watch lectures attentively. I will put it that way. Okay. Wasn't willing to make that sacrifice. But we've got 6 of us in here, and then one person online so far. Some people aren't going to make it today, and some people might be late today. So we're not going to wait on them though. This is, well, it's the cover of the book, but this is going to be my slides. We're going to actually lecture on the slides today. Let me tell you, I think we're all there right now. I don't know what it is. I don't know if it's changing seasons or what, but I was that way last Friday, like Thursday. And I don't know about you guys. I tried, okay? I tried to get us out before the storm, but I ended up driving through 20 minutes of hail. So, so it was the same because I was going, what direction is that? West. So I was going west, and so I was driving like basically into the heart of the storm, and it was 20 minutes. It was ridiculous. It was small hail. I didn't get any hail damage, but still, it was like my heart. I mean, my car's paid off. I was just like sitting there being like, but it was fine. You know, she made it through. I'd say she. Actually, my car has a name. I call my car Leroy Jenkins. So it's just all in or nothing, man. That's what I call it. All right, so we're gonna go get started. Just a reminder, these are our learning objectives. I'm not going to go through all the learning objectives. These are there more or less because these are slides I'm using for next semester, so I have them all prepped and everything. Just a reminder, as we're studying, when it says something like, explain the roles of cytokines in mediating the inflammatory response, if we were doing our concept map and we were addressing this effectively, then we would actually explain the roles of cytokines in mediating an inflammatory response, not just define what a cytokine is or saying mediation of inflammatory response dash cytokine, for example, right? So that's the spirit of what I would suggest studying, at least. Even if you're not doing it in your concept map, studying should at least be approaching actually being able to do what the learning objectives are suggesting at a level that you shouldn't have to refer to your notes to answer them, is what I'm saying. So that's the spirit, just a reminder as we're getting into the middle of this. I'm bringing it up because chapters 15 and 16, they're pretty hefty. So it's going to take some work to get to that point with these. All right, so our immune system is divided into 3 major lines of defense. The first line of defense is our barriers, essentially. So chemical, physical barriers, even the microbiota serves as a barrier against things. This would include things like the keratinized skin layer of your skin. When we think about our skin being keratinized, we often don't think about that. But then when you have somebody that has a burn, let's say somebody has a 3rd-degree burn, you burn past that keratinized, epidermis, the stratum corneum, if you will, of that keratinized layer of cells, and we've exposed the underlayers to microbes that we're worried about that person getting infection, because that's how effective that outer layer of skin really is at preventing infection and preventing microbes from basically getting into our bodies. So keratinized skin layer, your eyelashes, your nose hairs, things like this are actually preventing debris, including the microbes that comes with it, from getting inside of our body. Your saliva has enzymes in it that can break things down and prevent them from getting further in. Your stomach has acid. It's for digesting your food, yes, but also prevents microbes from getting into deeper parts of our body. So all of that falls into that first line of defense. Even though that might not be the primary function of stomach acid, it still provides a barrier against microbes being able to set up home. The second line of defense is whatever your inflammatory response, your complement cascade, fevers, your white blood cell, your phagocytes, what they're basically doing to prevent microbes from getting in generally. But once we start getting specific with specific antigens, then at that point, we are looking at the third line of defense. So we'll start breaking that down in chapters 15 and 16. All right, so we have a little question coming up. Drop a pin. Where on that chart would nostril hairs fall? Be general or specific. This is a little bit hard of a question to navigate on your phones, but do the best you can. We won't have a lot of them like that. Right, so I would say nostril hairs were in the first line of defense. That's that basic stuff, our physical barrier. It is definitely a physical barrier, but we're on this side. It's an innate. It's a first line of defense. It's a physical barrier preventing things from getting into that stuff. Our specific stuff is going to be our third line. Okay. All right, so we talked about some of this stuff, hydrochloric acid in your stomach, for example, other digestive juices in our digestive tract. Even semen has antimicrobial chemicals in it. And the vagina has a protective acidic pH that's actually maintained by the biota of the vagina that prevents some microbes from being able to grow. Lysozyme is an enzyme that we find in tears and saliva that actually breaks down peptidoglycan. That's what it does. And it's very general, type of purpose, but that's what it does. It is meant for breaking down peptidoglycan and is pretty effective at doing that. That's a part of our first line chemical defenses. Microbial antagonism, which we've talked about previously. Microbes that are part of your biome can take up stadium seeds. They can use the nutrients in the area. They can basically make antimicrobial compounds that prevent other microbes from being able to live in the area. They can also alter the local pH that might make that unfriendly to other microbes. And they also are really important in basically training our immune system on what, when, and where, and how we should fight microbes. So if we see intact gram-negative bacteria in our gut, then that might not necessarily trigger our immune response. But if we find that same gram-negative bacteria in your bloodstream, your immune response would get triggered. And part of that training happens in the gut and helps trigger like when and where and how we respond to microbes or parts of microbes. The genitourinary tract also has flushing of urine that can help flush out microbes in the area, and the vaginal secretions, which can also help with flushing out microbes and catching them in mucus so that they can't travel further into the reproductive tract. In the respiratory tract, we have trapping of microbes from the nasal hairs, copious flow of mucus throughout the entire upper and lower respiratory tract. And we have the respiratory tree where we have the ciliated epithelium, or the ciliary elevator, if you will. And the cilia in the respiratory tract constantly move mucus and fluids out of the lungs, and that's what you cough. Whenever you cough, you're helping that whole process eliminate what's in the lung. There are some diseases like whooping cough, or if you have a severe enough cough for a long enough period of time, this can damage your ciliary tree or your ciliary elevator to the point where your cilia can't remove things out of your lungs anymore. So then you not only have whooping cough, but you also are susceptible to secondary infections because your lungs can't clear those out either. So it can become quite a compounding problem. Talk more about whooping cough and stuff like that when we get into Unit 4, but this is leading into the importance of that, ciliary, movement to remove stuff out of the lungs. Mucous membranes obviously are more moist and permeable than like a keratinized skin layer. Mucous membranes are going to be more susceptible to infection as a result. However, we often have flushing of fluids in the mucous membranes, the mucus itself trapping the microbes. We can even have presence of lysozyme and stuff like that in the mucous membranes, as well as other antimicrobial compounds to protect those areas. We also have specific immunoglobulins that are targeted for the mucous membranes, which we'll learn about in chapter 16. Hair follicles can be shed. Skin glands can help make sebum and things like this that can be antimicrobial and also affect the ability of microbes to grow. Skin has that keratinized waterproof layer. Keratin itself is a waterproofing protein that helps adhere basically your skin cells together to create almost a sort of armor on our skin that protects us. That is called the stratum corneum. And that's pretty much the summary of our physical and chemical defense layers. Again, there's a little chart depicting a lot of that stuff and how it works if you need a refresher. So we're moving on now to our second line of defense and our third line of defense. So this is the rest of this chapter mostly focusing on that second line of defense. And we'll get introduced to some of the players of the third line of defense in this chapter as well. Immunology is basically the study of our immune responses. So our ability of our body to distinguish between foreign and self, and its ability to be specific about that as well. So destroying foreign, identifying things as foreign, and then targeting them for destruction. That's our immune system. White blood cells are the main key factor at play in our immune system. White blood cells are also known as leukocytes. Leukocytes. Our lymphocytes are a different type of leukocyte. So all white blood cells are leukocytes. Lymphocytes are a subtype of that. And then the lymphocytes include the B cells and the T cells. And we're going to talk about the different types of T cells and stuff like that too. Okay. So they are just a breakdown of the different types of cells. So all of our white blood cells are known as leukocytes. And many of them are trained to recognize self and non-self. This can be very general. So it could be saying like, I recognize people and I can tell that you are not a friend versus a friend, enemy versus friend. But can I tell that this is enemy, Duncan, or, enemy Evelyn, or enemy Jonathan, whatever. And can I tell that it is Evelyn's eyeball? Stuff like that. So then we would be getting into antigens at that point. Okay, so if I can recognize that this is Evelyn's eyeball, and that is the antigen side of things. But some of our white blood cells can just say, this is not my friend. This is non-self. So those general kind of target molecules, we'll talk mostly about those in this chapter. And we'll come back to antigens, our specific things that identify as Evelyn's eyeball. We'll talk about that in chapter 16. Okay? So any of these can be identified as antigens, whether they're very, very specific or very, very general. These are all going to be molecules that your immune system could identify as foreign. Okay. They're usually found on the surfaces of bacterial cells or any of the pathogens, some sort of marker on the surface of our pathogen that we can identify. Whenever we're able to tell the difference between self and non-self, sometimes that's an imperfect system, depending on what signals we're getting and what's happening in the body at all at the same time. So we might accidentally signal our body to say that self is non-self, and we might attack it. We're going to get very specific about this in chapter 17, but the general idea is that training that we have in place that tells our body that this is self, we don't attack self, and this is not self, we do attack not self, that's foreign stuff, that system can get crossed sometimes. And that can lead to autoimmune disorders like lupus and rheumatoid arthritis and type 1 diabetes. We'll talk about the specifics of that in chapter 17. So first things first, in the innate system, a lot of this depends on recognizing molecular patterns. So not saying this is Evelyn's eye, but saying the fact that you have, a shape of a human body means that you are a human. I only could say that. Okay, so really, really general stuff here. These are recognizing patterns and then being able to say, you fall into this category because of this pattern. So not a specific aspect of a pathogen, but the fact that it has a pattern. So what type of patterns might I be talking about here? Having endotoxin. If we see endotoxin, which is lipopolysaccharide, that's a pattern that our body can say, if you have that, you are not self. Peptidoglycan, found in bacterial cell walls. If you have that, you are not self. Okay. And then another one, double-stranded RNA. Our cells do not have double-stranded RNA. We only have single-stranded RNA. So if we see double-stranded RNA, that's something that viruses have. And so our body can say not self. Those are really obvious ones that our body, we can definitely say those are used as patterns to help our body recognize not self. So our cells can have receptors on their surface. Our white blood cells can have receptors on their surfaces that are called pattern recognition receptors that recognize those pathogen-associated molecular patterns, or PAMPs. You should know what a PAMP is, and we have some examples in mind, like peptidoglycan, endotoxin, or double-stranded RNA. We good? All right, so that's our first step of recognizing non-self. All right, true or false? A PAMP is a molecular marker that helps the innate immune cells recognize general groups of microbes. Is that true or false? Didn't bring my water. I should have brought my water down. Okay, so this one is true. This is the general statement of what a PAMP is. It's a molecular pattern that our immune cells can just say, you are not self. And we can even kind of basically use that as a categorization of the bad guys as well. So next thing is understanding our lymphatic system, where a lot of our immune functions occur. All right. And so a lot of our filtering and our checkpoints are going to happen in our lymphatic system. It's a drain-off system. So if you get an injury, like you stub your toe, right? This is a common example that I use in many of my classes. If you stub your toe, that's an injury, and we've got damage to cells. We're going to be sending out signals saying there's damage, and we're going to start calling in our inflammatory response. Basically, the whole idea is if you stub your toe and there's damage there, there's a potential for microbes to move into the damaged area and then potentially invade your bloodstream. Well, your bloodstream is an open stadium. It's a whole bunch of nutrients and cells and resources and a nice, warm, moist area for microbes to grow. There's no microbial antagonism, but there are your white blood cells and things like that in your blood. But still, it's a big risk to take for an open stadium like that. So we need to protect our bloodstream. We need to filter the stuff that's coming in from your injured area and filter it all before it hits your blood. That's what your lymph does. So anything that comes from an extremity that's in a tissue cannot enter your bloodstream until it has been filtered by the lymph. So your lymph basically works as its surveillance system that way. So lymph itself is like a plasma-like fluid that is fluid that's bringing fluid, cells, nutrients for the cells, signaling molecules and stuff like that. Like pretty much everything but your red blood cells can be found in the lymph. It's like your liquid portion of your blood. And it can all move out of the blood and into your tissues. Well, let's say we do that. You stub your toe. The fluids and the white blood cells and the clotting factors start moving out of your bloodstream and into the tissues. They cannot move back into the bloodstream. When it comes back, we have to filter it back through the lymph. And the lymph is a separate set of vessels, completely separate. And we have checkpoints along the way, including lymph nodes and lymphatic tissues, in order where we will have a concentration of white blood cells in order to do all the checking. Lymph only moves from the extremities towards the heart, just like your veins do, but it's a different set of vessels. And so it's completely and entirely driven by the contraction of your skeletal muscles. Your heart might pump your entire circulatory system. Your heart beating pumps the blood away from your heart with such a force that it forces the blood to move back towards the heart. And it just keeps that moving. But your lymph is a completely separate, closed off system from that. And so the only thing that can move lymph back to your heart is going to be your skeletal muscles. And as we're pumping it from the extremities back to the heart, we're passing it through your lymph nodes and then basically checkpointing them, just like we would at customs. So think of each lymph node as like a customs checkpoint. And so everything coming back in from your stubbed toe, from the dirt, the debris, the bacteria, the dead cells, and all of that stuff, including the neutrophils that went out there to eat things up, and all the fluid that moved out, comes back through the lymphatic system via your skeletal muscle movements. This is why it's so important whenever your patients have surgery or something like that. What's the first thing that we want them to do? Walk, if they can. And if they can't walk, then we want them to move somehow. And if they can't move, then we will put those things on their legs that squeeze their legs alternately. And that will help move the lymph for them. This is really important. This also gets us into that TikTok trend of lymphatic drainage, where you see people doing all these massages on their faces, trying to make their faces look slimmer. Now, the likelihood that's going to make your face look slimmer, I don't know what to tell you, okay? Do it if you want to do it. But does it help your lymph recycle back in? Absolutely it does. But is your face round because of that or because of lymph? I don't know what to tell you. Okay. Sometimes, yeah, okay. Yes, sure it is. Okay, so, but yes, is there any harm in doing it? No. So if you want to help your lymph move, give yourself lymphatic massages. There's no harm in it. It only helps your lymph. It does help with that. It helps your immune system. So good luck with it. But yes, so skeletal muscle movement and lymphatic massages, things like that are going to help the lymph get reincorporated and move and get pumped back towards the circulatory system where it can rejoin. Now, the idea is as we're doing all of that, it's getting checkpointed and we're checking all the debris and the cells and the microbes that could have been transported in the lymph at our checkpoints in our lymph nodes with all of that. Okay. We have another question. No, not yet, sorry. Here's a picture of the lymphatic system first. I thought it was next. I mean, we can see here that the lymphatic system is laid out very similarly to how your venous system is laid out, but it is a completely different, separate system with different vessels involved in it. In fact, if you have taken anatomy, you may have seen a few of the vessels. They can be large enough to be able to be seen. And you may have seen the lymph nodes and aggregation of lymph nodes in these certain areas. We get aggregations of lymph nodes, which is lymphatic tissue where we have a lot of our immune cells interacting in our groin and in our armpits and in our gut. That's the most common place, or in our neck too. And you can actually feel those lymph nodes whenever they do get swollen up. And we're going to talk about why they swell up as well, okay? But where else do you have lymph nodes? Everywhere. I mean, they're all over in your body, not just in the groin, in the gut, in the neck, in the armpits. We have them everywhere, all throughout. And they're dotted throughout our body, all along these vesicles. All right, so now the quiz question. Select all that apply. How is lymph transported through the body? I'm pretty sure it's two of the choices. All right, beautiful. So it is from the extremities back to the heart, and it's pumped by skeletal movement. Good job, guys. All right, our lymphoid organs, our primary lymphoid organs are the organs where we make our white blood cells and train them. Our secondary lymphatic organs are where they're going to be doing their job. Okay, so our white blood cells come from the bone marrow and from the thymus. So all of our white blood cells come from the bone marrow, but the T cells get extra training in the thymus. In fact, T cell stands for thymus cell, because that's where all their training takes place. But they all start in the bone marrow from the hematopoietic precursor cell, and then move into the T cells, move into the thymus for further maturation. B cells and the other cells, they do all of their maturing in the bone marrow. Okay. All right. Our B cells and our T cells often do their meetup and activation and stuff like that, interacting with antigens and everything like that in our lymph nodes. So it's like our basically police center. So police center, the police office, or whatever. So this is where we would take all of our bad guys in there and check them out and question them and things like that, as well as activating all of our squads and stuff like that would be happening in our lymph nodes. The spleen is another secondary lymphatic organ, but the spleen functions a little bit differently. While the lymph nodes and the salt, molten galt is often involved in basically filtering out the lymph, and the salt, molten galt is basically serving as an area for, like, the skin is the skin-associated lymphoid tissue, is salt. Malt is the mucosa-associated lymphoid tissue, and galt is gut-associated lymphoid tissue. But these serve as hubs for, like, white blood cells in those tissues. The lymph nodes serve as hubs along the lymphatic vesicle or vasculature, whatever you want to call it, those vessels of the lymphatic system. The lymph nodes are positioned there to filter out the lymph, but the spleen filters the blood. So we know that the spleen, or if you've taken anatomy, you probably learned that, or I don't know at what point you learn about the spleen, but the spleen is there as an organ to filter out your red blood cells, basically to take care of old red blood cells, damaged red blood cells, and things like that. But the spleen is also there to act as an accessory organ for the lymphatic system as well. where we're filtering the blood for microbes, not the lymph. Okay, we check in on that. So if for some reason it made it through your lymphatic system and got into the bloodstream, the spleen is there as like a secondary check to make sure that we're checking the blood. Okay. Can you live without your spleen? Yes, we know that you can. Will you be more susceptible to infections if you don't have a spleen? Potentially. Just depends on the infection in the setting. All right. All right. All right, bone marrow is where all of our white blood cells and our red blood cells originate from our hematopoietic precursor in the bone marrow. And then the T cells will move and mature in the thymus. The thymus is located about here in your chest, and it will shrink once you reach puberty. It pretty much starts shrinking and becomes less effective. Lymph nodes, again, aggregations of them found in the armpits, in the neck, and the groin, and the gut as well. These are just basically organs where we have a lot of interaction of our lymphocytes with antigen and activation of our lymphocytes in them. Now, a normal, healthy person that does not have an infection, anybody know how big a lymph node is? So normal lymph node, it's super tiny. It's about half, I mean, it's not even the size of this. I pulled the eraser as an example, but it's about this size, the eraser. Normal, healthy lymph node should be that size. Okay? So about half the size of a pea. Okay, size of a pea to about half the size of a pea. And if you ever work in transplant medicine, you will become very familiar with this. As somebody that worked in the transplant lab at Integris, I can assure you that this is something that we had to work with all the time. If a donor kidney, lung, or liver, or heart was coming in and we needed to test the donor to cross-match them with the recipient, we needed the donor's lymph nodes. And so the surgeon will not only collect the organ, but also different samples of the lymph nodes to be sent to each lab for testing for that purpose. We usually get sent anywhere from 1 to 5 lymph nodes in a little vial. And they are tiny, man. And we would have to flush them out with solution to try to get the B cells and the T cells, the lymphocytes, to come out so we could see if they would cross-match and react with, the patient's blood and stuff like that to see if there's any cross-reaction one way or the other. And so we often use that as a source of those cells. So lymph nodes are actually pretty small in otherwise healthy people. So they're pretty small. When you have an infection, your B cells and your T cells in your lymph nodes, they're multiplying by from 1 cell up to millions of cells. And so that causes your lymph nodes to swell as a result of that multiplying of those cells. Okay. Yes, that's what it's talking about. The spleen, we kind of already talked about it, and the associated lymphoid tissues, again, just areas of tissue that serves as a hub for our white blood cells. All right, blood itself consists of obviously red blood cells and then not red blood cells. This includes the liquid portion of your blood where your antibodies are found, your complement is found, any of your clotting factors, all that stuff, proteins that are found in the water part of your blood. That's what makes up plasma. So if I take your blood and I just collect it, just plain old blood, and let it sit, it'll clot, right? And so if I separate the clot from the liquid, that liquid portion is called serum. So if it's clotted blood, the liquid portion is called serum. If it is blood that I've treated so that it won't clot, the liquid portion is called plasma. Okay, we're tracking? If I add something so that the blood doesn't clot, then we call the liquid plasma. This allows us to separate out the different types of cells too through centrifugation. So it's where we spin it in the spinning thing, which we do quite a lot of in the lab, on the lab side of things. And it can even, if we add something to it to keep it from clotting, we can even separate out the red blood cells, and then the white cells and the platelets will sit on the top, and then the plasma. We can actually separate out the white cell layer, and that is called the buffy coat. In blood donation, this is incredibly important because this is where we can get white blood cells for donation and stuff like that. We don't always want just the red blood cells. We might want the red blood cells, we might want the platelets, we might want the plasma, or we might even want the white blood cells. It just depends. And so we can separate that out through centrifugation in blood donation. All right, the white blood cells themselves are known as leukocytes. Leukocytes, again, general term for all the white blood cells, all right? And we have 2 categories of white blood cells, granulocytes and agranulocytes. Here's our chart. Okay. These are named based on what they look like in a microscope. If they look like they have grains of stuff in them, granules, then we call them granulocytes. That's really it. That's how we define it. Okay. Our granulocytes are the fills, all of the fills, and the mast cells. So neutrophils, eosinophils, basophils. If it has fill, it's a granulocyte, plus mast cells. Those are granulocytes. The agranulocytes kind of fall into two categories. The monocytes and their two derivatives. A monocyte is a phagocytic white blood cell that is an agranulocyte. So it's a phagocyte also. It's able to eat things up, all right? But it's not a granulocyte. A monocyte is found in the blood, whereas if it moves into a tissue, it differentiates into a macrophage or a dendritic cell. So macrophages and dendritic cells were monocytes when they were in the blood. But once they're in the tissue and they're assigned to those tissues, they become macrophages and dendritic cells. And macrophages are probably the most important mediators in the whole immune response. We couldn't do most of what we do in our immune response without our heavy lifters, the macrophages. We'll talk about why in a moment, okay? Then we have the lymphocytes. The lymphocytes include our T cells and our B cells. So T lymphocytes and B lymphocytes. There are other lymphocytes. The natural killer cells, the natural killer T cells, and the gamma delta T cells that I am not going to talk about in this class, okay? So what I want you to focus on are the T lymphocytes and the B lymphocytes. I'm sure if you were paying attention when you made your concept maps, you know that that is enough to talk about, okay? So we'll talk about those guys. Okay. And just a reminder that our red blood cells and our platelets are not white blood cells. I mean, red blood cells obviously are not white blood cells, but they are non-leukocytes because of that, okay? All right, so that's our gist of our breakdown between our agranulocytes and our granulocytes. Let's have a little question about it. Select all that apply. Which of the following cells are granulocytes? All right, so the neutrophils are the only ones on here that are granulocytes. Sorry, I tricked you. But yes, it is just the neutrophils. Remember, monocytes and macrophages are kind of the same thing. The monocytes are the blood version, the tissue version is the macrophages. Lymphocytes are B and T cells. Those are all agranulocytes. Granulocytes are our fills and our mast cells. All right, so how do these white blood cells get around in our body? Obviously, they move via the blood, but once they get out of the blood and they need to migrate into our tissues, they need to be able to get to the surface of your skin and stuff like that to be able to fight off microbes in those areas. So how do they get from the bloodstream into our different tissues? They move around using the mononuclear phagocyte system. This is basically like a extracellular connective network tissues network that just is there to help the cells move around within the tissues. And that's what it is. It's like a highway between our cells within our organs. All right, so again, those monocytes are what are in the bloodstream. Once they move into tissues, they become macrophages and dendritic cells. And they're actually usually specialized to those tissues. So when we have macrophages that are in the liver, we call them Kupffer cells. In the lungs, we call them alveolar macrophages. In the skin, we call them Langerhans cells. In the brain, we call them microglia. So they can become specialized to those specific areas. So once they move out of the bloodstream. So again, monocytes change into macrophages or dendritic cells, and they can become even more specialized within their tissues and then have these specific names in those tissues. They're able to phagocyte, they are phagocytic. So basically they come and they recognize a PAMP, right? So that's how they're going to recognize things with their pattern recognition receptor. They come and they say, you have a pattern that I recognize as foreign. And now I envelop my whole cell around you and I eat you. So that's what our phagocytes are going to do. So monocytes, macrophages, and dendritic cells, and they're all kind of the same. They are phagocytes. We're going to talk about other phagocytes in a moment, including neutrophils. So whenever we have damage going on in our body, or whenever a phagocyte is eating something up or recognizes something as foreign, we can also signal with cytokines. And a cytokine is a cyto being cell, and kine is basically signal, okay? And these are signaling molecules that are products of monocytes, macrophages, dendritic cells, lymphocytes, fibroblasts, tissue cells that are damaged can also make some of these cytokines, mast cells, platelets, endothelial cells of the blood vessels. Pretty much any cell can signal in certain different ways for certain different things to happen. There's 4 major categories of our cytokines. Pro-inflammatory cytokines signal for the initiation of the inflammation response. Anti-inflammatory kind of signal for abatement of that, decreasing. Vasodilators or constrictors. What generally will happen if you stub your toe, we're going to increase blood flow to the area and decrease blood flow away from it. We want to increase the blood to that area and let all the cells and everything move in there, but we don't want the bad guys using your blood as a highway out of there. So we decrease the blood flow out of there. And then of course, we're going to reverse that once we have healed everything and we've taken care of the bad guys. And so those signaling molecules are important for vasodilation and vasoconstriction. And then we have the growth factor cytokines. These are signaling molecules that basically trigger our body to start multiplying more of your white blood cells. Okay. All right. Phagocytosis. We've talked about it a lot, and now we're here. We're at the place where we're going to get more specific with it. Phagocytosis is when your white blood cells eat up a bad guy. A lot of this stuff is triggered by PAMPs, pathogen-associated molecular patterns recognized by pattern recognition receptors on our phagocytes. Right. So you're a neutrophil or a monocyte or a macrophage. Dendritic cells also, okay, they also fall into here, but these are major types, okay? So neutrophils, monocytes, and macrophages are our biggies. And there's other ones that also fall into this. This is not an exhaustive list, okay? It's just our major guys. Your neutrophils are your most numerous of your white blood cells in your bloodstream. Let me see if this is the next thing. Yes, okay. The neutrophils are, and they can recognize a PAMP and say, you are foreign. Don't care what you are specifically. I don't care what part of a bad guy you are specifically, don't care, but I can tell you're foreign, and now I'm going to envelop my cell around you and eat you up. And neutrophils will eat it up and digest it and either poop out the waste or die as a result of eating enough things. That's pretty much all they do. They also will signal for more help to come in. They're our first line troops, okay? They get to the spot, they say, you're wearing, the wrong color, you're in the wrong uniform. We're all wearing this marker. You're not wearing that marker, you're foreign, therefore we're gonna eat you up and kill you. And they're very general about that. And they don't do anything else about it, except they'll signal for more troops to come in, but they don't care what you are. They don't care if you are Evelyn's eyeball yet, but they know that you're not self, okay? We haven't gotten to antigens yet, but we've gotten to PAMPs, pattern-associated or pathogen-associated molecular patterns that signal you're bad. And they'll eat up and destroy based on that. Almost any infection will trigger neutrophils to increase. So if you've got an infection, you'll have an increased number of neutrophils. If you have a parasitic infection, you'll have an increased number of eosinophils. So eosinophils are usually associated mostly with parasitic infections. That's what they're made for. But our bodies in first world countries were not trained to deal with parasitic infections at young ages like they were meant to be. We have not been on this earth for long enough for us to lose our identity of having parasitic infections early on in life, what we used to. So our immune systems aren't primed for that in the first world countries where we have hygiene. In third world countries, what you will see is that they don't have allergies because their immune system is trained properly to deal with those things. We'll talk about that in chapter 17 a little bit. So, okay, so eosinophils, they can go through phagocytosis, but they're usually triggered in response to parasites. That means helminths and protozoa. Okay, we don't have a lot of those here. Basophils are also associated with typically that sort of stuff as well, but they're a little bit less well understood and a little bit more complicated than that. All right, so that's our phils. Our monocytes and our macrophages, back to these guys. They are phagocytes that will, in the blood, be monocytes and transform into macrophages in the tissues. They will increase in size as a result of it. They'll develop lysosomes and other organelles that are associated with breaking things down because they are phagocytes. but the macrophages are generally scavengers of what the neutrophils left behind. So think of it as your first line troops, your neutrophils. You stub your toe, the cells in the area start sending out cytokines saying, help, there's damage here. The neutrophils are in your bloodstream. They hear the call for help. They follow the calls, the cytokines, to the area and start eating up anything that has a PAMP. You have a PAMP, eat you. Have a PAMP, eat you. Have a PAMP, eat you. You're not self. I'm going to eat you. I'm going to break you down. I'm going to leave behind your debris and my dead cells from having fought that. As we're doing that, we're also sending out signals saying, we're fighting a battle out here. Cytokines again. We're fighting a battle out here, cytokines. Then the macrophages, the monocytes, we should start with them, right? They're in the bloodstream first. In the bloodstream, they get those signals saying there's a battle happening. They come out of the bloodstream and into the tissues and turn into macrophages. Now they're listening for the calls of the neutrophils or following their signals of where they die, and then they're just scavenging, picking up all the waste of the neutrophils. But this is really, really important for what happens in chapter 16, because the neutrophils just left their debris around. But the macrophages are like investigators. So they're coming around and picking up all the parts. Some of those parts might be, Evelyn's eye or Jonathan's hair or whatever. All these parts of all these possible enemies. And they're going to save them up and bring them to the lymphocytes. And they're going to ask the lymphocytes, do you recognize this? And they'll hold up on a little part that we will get to, okay, on their little arm. And they'll say, I found this outside. Does anybody recognize it? And somewhere there is a cell that recognizes Evelyn's eye. And that one cell, one T cell or one B cell or whatever. In this case, one T cell that can recognize Evelyn's eye. It is the only T cell that can recognize that as Evelyn's eye. And it said, I've been waiting to see that eyeball. And that one cell will multiply up into millions and then go and serve your body in the fight as the T cell that as it does its job that we're going to learn about in chapter 16. We're getting there, okay? But the macrophage brought that to it. And so without the macrophage cleaning up after the neutrophils, the T cell might not ever see Evelyn's eyeball and be able to react to it. Okay, we're following? Interesting fact, tattoos. The reason tattoo ink stays in your skin, it is foreign, and your immune system knows it's foreign. Your macrophages traveled out, well, my macrophages traveled out of my bloodstream and into my skin and ate my tattoo ink, and it stayed there. These are actually macrophages with tattoo ink inside of them. Macrophages are not eternal. So if that macrophage that ate that ink there dies, another one will eat what it ate. And that's also why tattoos don't stay perfect over time, because the macrophages don't stay completely perfect in place. The better you treat your skin, the less likely you'll lose that, and it'll migrate to different places. But it's all maintained by macrophages eating up something foreign and staying in that spot to try to isolate it where it is. And it doesn't have a problem doing that because this isn't fighting back in any way. So it's good just maintaining it where it is. And that's how tattoos exist in skin. Pretty cool stuff. All right. We ready to go on? Okay. So this is how phagocytosis works. First things first, we get signals from cytokines calling there's damage. We need help. Help. So they, Warren stubbed her freaking toe on the dog bed again. So help, we've got damage. And so all those help signals, those cytokines, calling the cells in the bloodstream, the cells that are just flowing around in your bloodstream, they hear, oh. And so it starts following the signals to the site until they get there. That's chemotaxis. Okay, so that's chemotaxis. That's how our phagocytes get out of the bloodstream and into the tissues and to the site. Chemotaxis following cytokine signals. Then we get there and we recognize the PAMPs with our pattern recognition receptors, and we eat up the bad guys because of the PAMPs. Again, we just circle our whole cell around them and put them in a little vesicle. The vesicle that we put the guy into is called a phagosome. Phagosome. I know it says phagolysosome up there, but we haven't gotten that far yet. First, you put them in a phagosome. You have a separate vesicle in your cell called a lysosome. We will merge the phagosome with the lysosome to make the phagolysosome. That's like you have a bag of toxic ingredients sitting in your cells, in your macrophages, in your neutrophils, and whatever. And we're going to merge that bag and just dump its contents in to the container that you trapped the bad guy in. Okay. And then it digests the bad guy within 30 minutes. That's the destruction step. And then we'll excrete any of the waste products out that we don't need. In the case of neutrophils, we're excreting it. In the case of macrophages, we might also be calling out, anybody recognize this part, or this part, or this part, or this part, okay? That's phagocytosis. Again, this is all driven by pattern-associated molecular patterns like peptidoglycan, lipopolysaccharide, or double-stranded RNA. The phagosome is the vesicle or vacuole that we ingest the prey into. The lysosome contains all of our toxic things. These are antimicrobial chemicals. Things like lysozyme are found in lysosomes. That's fair. It is there. Okay. Remember what lysozyme does? Breaks down peptidoglycan. So that's in the lysosome. So are reactive oxygen species. Remember superoxide and stuff like that? We don't just defend against them. We use them as weapons too. And so we will load up a lysosome with all this toxic stuff and then just merge it with that bad guy and say, good luck, buddy, have fun with this. And then, hopefully it breaks the thing down. And it often does. It's pretty effective. It causes death of bacteria within about 30 minutes. All right, here's a little quiz. See if you're paying attention. What is a phagosome? You should be careful choosing your choice here. This is the brown, I guess. I don't know. I guess it's a brown color. Looks brown to me. It's probably supposed to be yellow or orange, whatever. But yes, it's the vesicle that's created when the phagocyte ingests the foreign thing. The lysosome contains the toxins. When we merge the lysosome with the phagosome, phagolysosome. Okay. Okay. All right. All right. We have stubbed our toe. We're sending out the cytokines. And we're calling in the white cells, like our neutrophils and our macrophages and stuff like that. While all this is happening, remember cytokines are pro-inflammatory, or they are vasodilators and constrictors and all that too. So this leads to increased blood flow into an area. When you have increased blood flow, what color is blood? Red. So why does your arm, whenever you scratch it or whenever you stub your toe, turn red? Because of the increased blood flow. So rubor is the redness from the increased blood flow. It also leads to increased warmth because your blood is warm and your extremities are colder compared to the inner of your body. So when we increase blood flow to an area, we increase the temperature. That's calor, okay? Tumor is the swelling. So when we increase blood flow to an area, we're bringing the blood there to bring the cells into the tissues, right? So they can come and fight the good fight for us. The blood is bringing the cells as well as clotting factors, as well as fats and things like that that are needed to help power all of this, and all the other signaling molecules as well. So the fluid moves out of the blood and into the tissue. That leads to swelling of the tissue. That's the tumor, the swelling of the tissue from the injury. And then dolor is the pain. Probably the most important thing to let you know that something is wrong in that area. Sometimes helpful, sometimes not, we know. But without it, you would probably die. And we know this is true because people that have a reduced pain response, and I mean like if they don't have a pain response, tend to die much younger because they don't know that there's something wrong in their bodies. So pain is actually a lot more important. We take it for granted, I feel like. But all these things are signal and they're part and they have purpose in the whole cycle of letting your body know there is injury and that we are repairing it. And in bringing all of the things that are important for repairing it to the site. So those are the terms. Obviously, you need to know the terms. They're bolded in here for a reason, okay? Chronic inflammation is a problem because inflammation isn't just there only to be all good, but we also have destruction of tissue as part of the inflammatory process. We need to destroy damaged cells that were involved in the process, but we can also have some loss of healthy cells in the process. So chronic inflammation can lead to problems, often relating to damage in blood vessels and lungs and skin and joints as a result of this being going on longer than it should. Some of your hormones favor inflammation and some of them favor abatement of that inflammation. Obviously, cortisol is probably one of the biggest ones that is pro-inflammatory. And so having issues with stress can actually lead to increased inflammation in the body. For example, Okay, what is inflammation doing? We're attracting the cells that are going to repair things and eat up the bad guys to the side. We're setting in motion that mechanism of repair and getting rid of microbes, destroying them, and preventing the microbes from spreading to other areas, increasing the blood flow to bring in our white blood cells, to bring in clotting factors, to bring in our healing molecules, bring in complement and all of this other stuff, but then also impeding the flow of blood out of the area. So we're keeping everything confined. We're also going to wall off sometimes our infection with our white blood cells. We can do that as well. So we're mobilizing and attracting our immune components. We are repairing the tissue damage and we're destroying the microbes to block further invasion. All right, so here's a quiz question about it. What are some benefits of inflammation? What are helpful functions of inflammation? Don't read too much into this, okay? It's not meant to be a trick. This isn't all of the above on this one. Okay. All right, next. Here is a breakdown with images of what I'm talking about. So you step on a nail here. The nail brings in microbes. The damaged tissue, the mast cells that are in the tissue, and other immune components there are signaling for help. They are sending out cytokines, calling in your neutrophils. The neutrophils are coming in, and so is the fluid that comes in with them. So the neutrophils are coming in, fluid is coming in with them. We're bringing in complement. We haven't gotten to complement yet. We're coming to it. I know I keep saying it, but we're coming to it in a moment, okay? Other factors that are in the blood for healing and for basically targeting all this stuff and getting this whole immune reaction. This is all coming in with the fluid and the neutrophils coming as well. Then we have pus formation. Pus is mostly neutrophils. It's almost entirely neutrophils, but it's also bacteria, the waste components of fighting the bacteria, and the fluid itself that was brought into the area. So that's going to lead to the development of pus. As the neutrophils are eating up all these bad guys and they're leaving the waste all behind, and the fluid is built up in the area, and we're clotting off the bleeding and all of this stuff. Then finally, after calling out for help, our macrophages have moved in. They're eating up and getting rid of those neutrophils that we're fighting because we don't need them anymore. And we're picking up all the waste and scavenging all the waste. We're healing that tissue, and we have development of scars and scabs and things like that. Okay, we good? That's the idea. So the earliest changes in inflammation are going to be vascular changes, increase of blood flow and stuff like that, as well as transmission of all the signals and stuff that's involved in all of that, in order to start moving in the cells and the clotting factors and complement and all that stuff for that. The fluid, when it moves into the tissue, out of the blood and into the tissue, that fluid is called exudate. Once it gets reabsorbed back into the lymphatic system, then it's lymph. Okay, so it just changes based on where it is. So remember, it comes out of the bloodstream into the tissue where it is exudate. The swelling that you would observe as a result of that is called edema. That's what edema is. It's a movement of the fluid out of the blood and into the tissue. Remember, that fluid does not rejoin the bloodstream. It goes back into the lymphatic system, where our lymph nodes will be filtering it with all the white blood cells before it can rejoin the blood. So that's the idea of how all of this is going to work. All right. How do our white blood cells get out of our bloodstream if they're in our blood vessels? And that's a closed system. How does the fluid and how do the white blood cells get out of the blood vessels and into the tissues? Like our neutrophils have to, okay? And our macrophages have to. They move between the vasculature cells. So quite literally, their cells abutted up against each other, and the white blood cells are like, excuse me, and they push their way through. And this process is called diapedesis. And they force their way through those junctures in your tiniest capillaries, where it's like one cell thick, one-to-one cell, and they can just push their way through. All right, so that's diapedesis. Microbes can push their way through too. And so this is why we have the blood-brain barrier. So the blood-brain barrier, this is your tighter junction. So less things are able to squeeze through. And there's going to be less availability of those junctions where you could squeeze through. So this protects your brain from dangerous molecules, from dangerous cells, but it also means that we can't get our white blood cells there as easily. So we typically have our own sort of segregated immune sort of response in your brain and in your eye as well. We call them immune privileged sites. This is because evolutionarily speaking, cavemen couldn't afford to lose their vision. And obviously, if you have damage that can be done by inflammation, that can damage your tissues, what happens when your central nervous system is damaged? Can it repair itself? Nope. When you're paralyzed forever. And so the body cannot risk having your brain tissue damaged from your immune response. It is not a perfect juncture, but that's the purpose of the blood-brain barrier. We're keeping microbes and stuff out with it. We're also keeping our own white blood cells that could kill our central nervous system out too. So we have our own sort of like backup plan for our central nervous system. Okay, so our immune privileged sites, we'll learn more about that in Unit 4. The ability of our white blood cells to cross over from our bloodstream and into the tissues and squeeze through, that's diapedesis. Next thing is a question about it. What's it called? What's it called when the white blood cells migrate into the tissue from the vasculature? What's the term for it? Here's a guy who's doing it. He's diapedesising through a crowd. All right, so diapedesis. Good. All right, so we need edema, we need chemotaxis to bring our white blood cells to the site and all of our clotting factors and things like that that are involved in healing. It also helps dilute out any toxins and stuff and that might be produced by bacteria and things like that in the area, and brings in that whole, like all the healing molecules for it. Eventually, after a long time, after we do our basics of calling in the inflammation response, our macrophages and our monocytes and our lymphocytes get called to the area. We can start clearing out what the neutrophils cause and collecting our evidence to call in the appropriate lymphocyte to the spot. Again, we found Evelyn's eye. We want the cell that recognizes Evelyn's eye. We'll multiply that guy up and create our own specialists out of that. Okay. All right. The part of the inflammatory response, we start talking about calor and the increased warmth. That can be a localized thing from increased blood flow, but we can also increase the temperature of your whole body. And that can also have benefits for us as well when we're fighting, guys. So this leads to fever. Fever is basically low grade, moderate, or high. We do not consider anything below 100 degrees to be a fever in an otherwise healthy person. Now, if you are a geriatric patient who is basically like low muscle tone, and it's basically sedentary, then we might not consider 98.6 to be a healthy temperature either. And you guys will learn about that more when you get into nursing school or wherever you're going. But that's the idea here. This is kind of depends on your scenario, your patient, and stuff like that. Generally speaking, 98.6 is normal temperature. 100 would be considered a fever. 102 is moderate. 104 is high. All right. Anything that can cause a fever is called a pyrogen. Think pyro being fire, right? And so increased in temperature. So pyrogen, things that generate pyro, fire, okay? And so these can be exogenous from outside of the body or endogenous. These are signals sent by your own cells. So if your neutrophils are fighting a lot, if your monocytes are fighting a lot, if your macrophages are fighting a lot, they can release things like interleukin and tumor necrosis factor as cytokines that basically signal we need to have a fever. It's time. We have seen enough PAMP. We know fever needs to happen. So that would be your cells. That's the signal coming from your own body. But there are other markers on some of our pathogens that can just immediately incite fever. Endotoxin is the most obvious one. LPS can immediately signal for your body to create fever. But then, and these can be any sorts of molecules that can be found on any various pathogens. Well, so what do we call a pyrogen? Oh no, what's the question? Which of the following are exogenous pyrogens? Pyrogens that came from outside of the body. Hint, it's only one of them. I know it's a multi-select, but I'm gonna help you out. It's only one of them. So here, cytokines are signal molecules made by our own cells to signal our own cells to do things in our body. Here, endotoxin is the only one that came from not our own cells to signal for this stuff, okay? All right, what are some of the benefits of fever? Fever inhibits microbes from growing by causing our body to hoard iron away from them. It also slows their enzymatic reactions. It's too hot for them to live. We can manage to have the heat increased. Number one, it speeds up our immune cells. Now we feel tired and we feel exhausted because we're putting so much energy into increasing our body temperature, because we're increasing our inflammatory response. We're putting all of our energy into that. And so we get tired from that, right? But the idea is that if we can increase our white blood cells function, they're going to fight off this infection. We'll feel better eventually for that instead of dead from the pathogen surviving and winning, okay? So it will increase our metabolism. It will increase our white blood cells' ability to find their way to the site of the infection, and increases the growth of our lymphocytes and the target of our lymphocytes. Again, we only have one cell that recognizes that antigen. We need to find it, and it needs to multiply fast. And we can speed that up with the signals involved in fever. So there's so many benefits for fever. Obviously, your body doesn't have a stop point. So if we keep signaling for fever, more fever, more fever, more cowbell, that's what it makes me think of. We want more fever, okay? Your body will keep delivering. So it will, if it gets enough fever and it's succeeding, then it knows, don't keep raising the fever. But if it's raising the fever and it's not succeeding, it'll keep raising it. And it doesn't know to stop. So it will raise it to a point where you can actually start denaturing your own proteins, first usually in your brain. And so your brain's pretty sensitive to that. So whenever we start having it be too hot, you'll denature proteins in your brain, and that can lead to brain damage as a result of that. So that's why we have to be mindful of those high fevers, okay? So there's a question about benefits of fever. Which of the following are benefits of fever? Again, don't read too much into this. We're getting there. You guys are doing great sitting through my spiel. All right, so this is an all the above again. All right, so all of that is. All right, should we treat fever? Fever has benefits. It has a role for us. Should we treat it? If you are really uncomfortable, treat it. If you're not really uncomfortable, if you're just tired and achy a little bit, but you can make it, and you've got a fever that's less than 104, let it ride if you're an adult. If you're a kid, always consult the pediatrician, right? We're just to be real. It's a little kid. You can make that judgment as a parent, and you and your doctor can make that judgment together. I'm not going to make the choice for you. But if it's an adult and you're otherwise healthy and you got the flu or something like that, and your fever is 102, let the fever ride. It's there for a reason. It's doing you benefit. Now you know. It's not just there to be a jerk. It's there to help you fight the microbe. Let it do its job, okay? Talk to the doctor where appropriate. What's the best medication, the safest medication for treating fevers? Tylenol, acetaminophen. All doctors can agree on that. Doctors. That's all I need to say, right? Okay. All right, moving on. Interferon. Interferon is another type of cytokine. Again, any signaling molecule, we call it a cytokine. And it's for self-cell to self-cell communication. So this is interferon is a cytokine that basically, like, if I am infected as a cell, I can send interferon as a little letter to my neighbor. And now my neighbor knows I'm going to make other molecules. I listened and I heard your interferon call. So now I'm going to start putting things in place to degrade nucleic acid, to block viral replication, and things like that. So antiviral mechanisms. So interferon signals that your neighbor is having something going wrong inside of them, and that you need to prepare for that. And so some of this stuff, should we as a cell be degrading nucleic acid? No, but if you have to save yourself and your other neighbors, you will. So this is like, it's the purge, okay? The purge is happening like in the movie, okay? All right. The purge is happening and people are coming around to houses and they're killing people in the houses to steal the things they have in the house. And you call and you say, they're at my house. And you're the neighbor and you know they're probably coming to your house. You're going to load your shotgun. And you might go down fighting to protect your other neighbors too. That's the idea of interferon. Okay, so interferon interferes with the pathogen's ability to succeed. Interferon. It's for viral infections, but we have also hijacked this ideology for cancer as well. So it's another mechanism that you can tell your body, I can give you infusions with your chemotherapy. So when you get chemotherapy for treating cancer, it's not one drug. It's usually a cocktail of different drugs. And many times those cocktails include immune factors like interferon to help your body start being like, we need to kill, there's an inside thing going on and we need to kill things with bad inside things going on. Does it affect your healthy cells too? Yeah, it does. Sometimes you're willing to pay that price though, if you're gonna go down in flames. So you might be willing to have some damage so that you can survive overall. So that's the idea. Okay. When we think of something like this, we're just saying, if I'm pumping you with interferon and your whole body is getting signals saying we need to degrade nucleic acid, suddenly now your cells aren't replicating and replacing the old cells. That's why your hair falls out, because many of those chemicals work this way. They inhibit cells from being able to multiply. All cells, not just cancer cells. And so that's the price you pay with chemotherapy, right? But the idea is the same here. You tracking? Interferon. Now we're at complement, which is at the end of chapter 15, okay? All right, the complement system. It's a whole slew of about 50 different proteins that just always exist in your body. We don't need to trigger them to be made. They're always there, and they're just waiting like mines. So I always think of like an ocean, like you always see, I saw this meme on Instagram, and it was like when you're a kid and you watch all these movies or you see all these stories about like all these mines floating in the ocean from like World War II, and you could hit them at any point with your boat and just set off these mines. And you get so prepared in your mind. When you're an adult, you're going to see all these mines in the ocean. You could hit them at any time, blow them up. I don't know. Anyways, obviously not real, right? But as a kid, you're treated like this is a constant threat that's in the ocean all the time, is these mines that could potentially be floating around. I think of complement as like that. They're like proximity mines, okay? But what they can be activated by are three separate things. So it's a whole slew of proximity mines out there, and there's three major things that can set them off, okay? And once you set one off, they're all going. You set one off, it's a cascade reaction. One boom, and it causes the neighbors boom, and it causes the neighbors boom. And all the parts of them can come together and create new mechanisms to fight off bad guys. So that's how complement works. All right, are we ready? Complement can be activated by three things. Three groups of things, I should say, can set off the bomb. The first is antibody. If antibody binds to something, bam, set off a bomb. That's the classical pathway of the complement pathway. The lectin pathway. If a protein called lectin, don't worry about it, okay? It's a protein called lectin that's in your body. If it binds to mannose, which is a sugar found on bacteria, we don't have it. If lectin is found binding to mannose somewhere, boom, you set off a bomb. Complement's activated. The alternative pathway. If we see LPS or other molecular patterns, they sound like PAMPs because they're like PAMPs. But remember, a PAMP officially is recognized by a pattern recognition receptor on a phagocyte. We're not talking about cells here. We're talking about a protein. So complements are proteins. If those proteins interact with LPS or PAMP-like molecules, pattern molecules like LPS, then boom, we set off a bomb. So we can recognize the patterns like LPS, alternative pathway. We can recognize lectin and mannose binding together, boom, lectin pathway. Or we can recognize antibodies binding to something, boom, classical pathway. Those are all different ways that we can activate the complement cascade, okay? All right, which complement pathway is triggered by antibody? We're almost there, folks. We almost made it through one of the chapters. But tell me this stuff isn't cool. It's so cool. And it's only cooler with chapter 16. All righty. More complicated? Yes. Okay. But also cool. All right. Once we set off a bomb, that's the initiation step. Okay. You set off a bomb. One of those three pathways. Then one bomb explodes, causing the neighbors to explode. That's the amplification and cascade, okay? Then we have polymerization, taking the parts of the bomb. Now that the bomb has exploded, those parts are like transformer almost. The parts can come together and bind with each other and do things. It has several things. Complement can do 3 major things. I don't know why I don't have a slide on the 3 major things, but it can do 3 major things. It can act as a molecular handle for phagocytes to grab onto. So after we've made our bombs explode, those parts can come together and our phagocytes have something to grab onto on the surface of our pathogens. You had that sticky coating of the capsule? Oh, so sorry. The complement system made new handles for our phagocytes to grab onto, and now we can eat you up. Good try, though. Good try. So that's called opsonization. And we will see it again in a little bit. Okay? So we can opsonization, we can trigger the inflammatory pathway, and we can also create the membrane attack complex. So we take the parts of the bomb and they can come together to make handles. We can take the parts of the bomb and they can signal inflammation. And we can take parts of the bomb and build the membrane attack complex. So what the heck is a membrane attack complex? It pokes holes in the pathogen. So it takes the parts of the bomb and literally builds a hole in the surface of the bad guy. That's bad. We have membranes on our cells for a reason, and so do our bad guys. They have coatings and membranes and stuff for a reason. So if you poke holes in them, they can't regulate what goes in and out of their cells, and literally they leak their contents and die. So the membrane attack complex creates holes in them to literally cause them to leak out and die. There is a video about this. It is 7 minutes, so we're not going to watch it. I apologize. But it is in the lecture recording on your website if you want to watch it. Okay? That's chapter 15. Do we have questions about chapter 15 before we move on to 16? I think we've set it up pretty good. We have a pretty good understanding of where macrophages are coming from and what they're doing. We're going to kind of review it a little bit when we get into 16. Okay, we're going to get a whole new Kahoot going. Yes.
Actually, just to be clear, cytokines, they are particles produced by macrophages and monocytes to release signals.
Yeah, so cytokines are the signals.
Okay, they are.
Yes, yes. So they are